Hello guys, welcome to Ferdinand tutorials. I hope you're doing well. Now, in this video, we'll be looking at biochemistry of nucleic acid. In this biochemistry of nucleic acid, we try as much as possible, to look at the meaning, types, and functions of nucleic acid. We look at the components of nucleotides, and lastly, we'll look at the differences that exist between DNA and RNA. Now, let's start with the meaning, types, and functions of nucleic acid. First of all, when we're talking about nucleic acid, we say that nucleic acid are long-chain polynucleotides. Now, this long-chain polynucleotide has specific functions. Some of the functions include we use it to transfer characters from parents to offspring. That is, we use nucleic acid in the formation of genes. Secondly, nucleic acid can also be used to form proteins through transcription and translation. Look at what I'm saying. For us to form a protein in the body, we have what we call this movement. Now, if we look at this movement, for a protein to be formed, we are going to first of all form RNA. RNA will be synthesized from DNA, and then the RNA that was synthesized from DNA, we use it to resynthesize protein. Now, apart from those two functions, DNA is also used in forensics. Now, in forensics, what they usually do is that they can use it to, detect a criminal through DNA fingerprinting. They can also use it to conduct what? Paternity tests. So those are some of the functions of nucleic acid. There are many more. Now, when we're looking at some of the types of nucleic acid, we generally have two types of nucleic acid. When we have the deoxyribonucleic acid, which is DNA, and then the ribonucleic acid, which is what? The RNA. Now, if we come here, we say we have DNA and RNA as the two types of nucleic acid. Remember when I was defining or explaining nucleic acid, I said that they are long-chain polynucleotides. When I mean that they are long-chain polynucleotides, poly simply means many, and then nucleotides is actually what? A component. Now, what I'm trying to say is that we have nucleotides that combine in a varying amount for us to form what? Nucleic acids. So, in other words, we can say that nucleotides are monomers of nucleic acids. I don't know if that is making sense. I say, in other words, we can say that nucleotides are what? Monomers of nucleic acid. Remember that we have what we call protein and amino acid. Just as I said that amino acids are monomers of protein, nucleotides are also monomers of what? Nucleic acid. That means nucleotides combine in varying amounts for us to form what? Nucleic acid. That was why in my definition I said that they are long-chain polynucleotides. That means we have long chains of nucleotide that combine together for us to form what? Nucleic acid. Now, when we're talking about the components of nucleotide, which is the monomer of nucleic acid, I hope you're following. Now, when we're looking at the components, we have three major components of nucleotides. We have, first of all, we have what we call the nucleobase or nucleobases. We have the pentose sugar. We also have the phosphate acid moiety. Now, let me start with nucleobases. Now, when we're looking at nucleobases, remember we're still talking about components of nucleotides. These are the three major components of nucleotides. What are nucleotides? Nucleotides are monomers of nucleic acid. What are nucleic acids? Nucleic acids are long-chain polynucleotides. Hope you're following what I'm saying. That means we can't have nucleic acid without what? Nucleotides. So, now, in nucleobases, we usually have two types of nucleobases. We have what we call the purine base and the pyrimidine. Now, when we're talking about purine and pyrimidine, pyrimidine has a one ring. They has a one ring structure. While the purine basis has what? Double ring structure. For my purine, I have the example here. I have adenine, I have guanine. You can see it as Ag. That's the two purine bases. Now, for my pyrimidine, I have cytosine. Are we there? I have uracil. I have thymine. So, in other words, I can call this CUT, cut. So, the nucleobase, which is the first component of nucleotides, contains what? The purine and pyrimidine. And then I said that the purine has two ring structure. When we're talking about two ring structure, two ring structure simply means we're having something like this, and then we have another ring, Are we together? This is a different ring. This is another ring. This is how a purine base look like. When we're talking about pyrimidine, pyrimidine is just one ring structure. Are we together? This is more like how a ring structure looks like. So this is a pyrimidine base. This is a purine base. Purine base has two ring structure. Pyrimidine one ring structure. Are we together now? Now, if we've understood this as the nucleobases in nucleic acid, I want us to look at the next component of nucleotide, which is what? Which is the pentose sugar. When I'm talking about pentose sugar, what I'm saying is that we have what we call what? The 5-carbon sugar. In carbohydrate, we have what we call 3-carbon sugar, which we call trioses. An example of the trioses, we have what we call glyceraldehyde. We have 4-carbon sugar like tetroses. Those 4-carbon sugar, example, we have what we call erythrose. We have 5-carbon sugar, which are 5-carbon sugar, which we call pentoses. Example is what we call the ribose and deoxyribose. We have 6-carbon sugar, which we call hexoses. Example, we have glucose, we have galactose, we have fructose. So we are not looking at all those 4-carbon, 6-carbon, 3-carbon. No, what we are looking at generally here is what? The 5-carbon sugar, which is the next component of what? This nucleotide. So in 5-carbon sugars, we usually have what we call the ribose sugar, and the deoxyribose sugars. I hope you're following. I want you to understand the component. Later I will come to explain the differences that exist in DNA and RNA, and then you understand, the usefulness of each of these, and the one that is found in DNA and the one that is not found in DNA. Please, if you've not subscribed to this channel, please click on that subscribe button. We need to grow this educational platform. It will be important if you watch our video, you try as much as possible to subscribe. It will support us. Are we together? It will make us to continue making more and better videos for your consumption and the consumption of others too. Let's go back to the video. Now, having understood the pentose sugar, I want us to look at the phosphate acid moiety. Now, the phosphate acid moiety is more like, the phosphate group that is added to this. We call it the phosphate group. Are we together? That is added to this. Now, what I'm trying to say is that these three things combine to form a nucleotide. Now, meanwhile, I can have nucleobase plus pentose sugar plus phosphate acid moiety. I'll form a nucleotide. What are the examples of nucleotide? Examples of nucleotide, we have what we call ATP, adenosine triphosphate. How is it formed? For adenosine triphosphate to be formed, that means a base must be introduced. What is the base I'm looking at here? I'm looking at adenine, which is a purine base. Are we together? Now, when adenine reacts with sugar, it forms a nucleoside. That's what we call nucleoside. For us to have a nucleoside is formed when we have a nucleobase react with a pentose sugar. That's how a nucleoside is formed. A nucleotide is formed when we have nucleobase plus pentose sugar plus phosphate group. The nucleotide will be formed. So this ATP is a nucleotide. It is formed when we have a nucleobase react with a pentose sugar plus what? A phosphate group. That's how ATP is formed. Are we together? So I'm saying that I have adenine plus sugar plus 3 molecules of phosphate group. That's why we have triphosphate, 3 phosphate groups. I don't know if you're getting it. So ATP is formed. Meanwhile, when we're talking about nucleosides, I said that they are nucleobase plus pentose sugar. So examples of nucleoside, we have what we call, when we have adenine plus a sugar, we form adenosine. That's a nucleoside. When I have guanine plus a pentose sugar, I'll form guanosine. Remember, base plus sugar is nucleosides. Base plus sugar plus phosphate group is nucleotide. Nucleotide, don't forget. So I'm still giving examples of the nucleosides that we have. So we are done with the purine. Adenine plus sugar is adenosine. Guanine plus sugar is guanosine. Now cytosine plus sugar is cytidine. Uracil plus sugar is uridine. Thymine plus sugar is pyrimidine. I hope you're following. So we are done with nucleosides. Now for nucleotide, if I take this adenosine and then I add it one phosphate group, I'll form a nucleotide. That's when I'll have adenosine monophosphate. So adenosine plus a phosphate group will form a nucleotide. So I can add two phosphate group, which is diphosphate, adenosine diphosphate, guanosine monophosphate, uridine triphosphate, uridine diphosphate. So those are how nucleotides and nucleosides are formed. I hope that makes sense now. Now let's look at the differences that exist between DNA and RNA. So when we're looking at the differences that exist, I want to say something. Now, in DNA is double-stranded. Are we together? Something like this. This is a typical structure of a DNA. It's double-stranded. Now, in single-stranded for RNA, single-stranded simply means that RNA is this way. While DNA, which is double-stranded, we are looking at something like this. Are we together? This is double-stranded DNA. This is single-stranded what? RNA. Now, the difference that exists between this, apart from this one being double-stranded, this one being single-stranded, bases that are formed, remember these are nucleic acid. And then we say that nucleic acids are polynucleotides. That means nucleotides combine in varying amounts to form this. Bases that are found in this DNA include A, G, T, C. These are the 4 bases that are found in DNA. Meanwhile, for RNA, bases that are found in RNA, we have A, G, C, U. In other words, for DNA, thymine is replaced with uracil in RNA. You understand? Why AGTC is present in DNA, AGCU is present in RNA. That means thymine is replaced with U in RNA. We say that DNA is double-stranded, RNA is single-stranded. In the basis, uracil replaces thymine in RNA. Are we together? Those are the difference. Another difference I also want us to understand is that in the sugars, remember I said 5 carbon sugars. When I'm trying to form my DNA, I uses the sugar deoxyribose. Are we together? In my RNA, I uses what? Ribose sugars to form my nucleotides. Are we together now? Now, I want to also say something. That is what we call, base pair rule. In base pair rule, we are saying that adenine bonds with thymine with 2 hydrogen bond. Cytosine with 3 hydrogen bond bonds with what? Guanine. Are we together? Remember that instead of this nucleic acid are bases, nucleobases that combine together to form this particular what? Nucleic acid. Are we together? We've looked at the differences that exist between DNA and RNA. Another thing I also want us to look at is that, in DNA are complementary to each other. That means here we can start with 5 prime and then we end with 3 prime. Here 3 prime and end with what? 5 prime. They are complementary to each other. Meanwhile, in RNA are completely, not complementary with each other. Since it's single-stranded, not double-stranded. So what happens is this. In A, we usually say that from base pair, we say that the concentration of A will necessarily be equal to concentration of T. I'm talking about DNA now. C, the concentration of C will be necessarily equal to what? Concentration of G. But in RNA, we usually say that the concentration of A, because it is not completely complementary, that is, it has 1 strand. But the concentration of A is not necessarily equal to T. The concentration of C is also not necessarily equal to G. Are we together? So those are some of the differences that exist between DNA and RNA. I hope I'm able to explain some of the things that you are having issues with, and I hope I explained this, biochemistry of nucleic acid to your very own understanding. If you have a question, or if you like me to make updated videos on nucleic acid, maybe we want to begin to look at more of the transcription, translation, reverse transcription, prion proteins, would like me to say 1 or 2 things about it, let me know in the comment section. I will make video. If you also want to support us, you can share this video to your friends, cause them to also learn as you have just learned. Don't forget to subscribe. See you in my next video. Bye for now.